how we want to do it. First, I will have not very long, I think it's about 20 minutes or something like this, PowerPoint presentation, and tell the main part. After that, you will allowed to have any questions you want about the project, about anything I say. To have a question, to ask a question, please raise your hand. Not you can raise your hand on camera, but it's better to raise your hand in Zoom. I believe you all know how to do that. And in our conference, we have a moderator. It's our coordinator Mishesh, and I believe Suresh will help him. They will monitor your raised hands and give you a possibility to speak when you will do that. So once again, I'm very glad to see all you guys here online. Now, to be honest, at first, when we started this project, oh, I should record the meeting, sorry. To be honest, at first, when we started this project, we haven't planned to make such educational seminars like now, but because of pandemic situation, we have to do it online. Firstly, it was planned for me and our team to come to your country and to tell you about what we are going to do, to teach you how to like deal with molecular methods offline. And we are still planning to do that. But because we cannot do it now, we should first teach some theory to speak about what are we going to do, and then I think it's still necessary to come to your country and to teach you how to extract DNA, how to deal with samples and everything like that. Now our project already started, and we are in the progress in the middle of buying some supplies and some equipment for each country to have a possibility to perform, to make all works which included in project. So I will start my presentation. One second. Yes. Okay, I believe you can see the presentation, right? Yes, I hope so. I will, if something wrong, please use chat, like text chat. I will try to monitor chat, and if something wrong, you cannot see something or anything like that, you just can write down on the chat and I will monitor it. Okay, so our project is called Adaptation and Implementation of Modern Molecular Biology Technologies in the Practice of Agricultural and Wastewater Treatment of ASEAN countries. This is our first educational seminar in a series. I'm thinking it's going to be 3 or a little bit more, or maybe 4 seminars at all. Today's seminar will be devoted to pathogenic microorganisms in wastewater, in water from agricultural objects, and methods of their detection, right? Because it's very important to detect like microbiological contamination in any water. It's especially important in drinking water, but in agricultural and treated wastewater, it's important as well, because this water goes directly to the nature, and a lot of people in ASEAN countries, in any country, communicate with that treated water. So it could be dangerous if it's not treated properly. First of all, we should speak about like what possible could go wrong. Yes. The first of all, we should discuss the possible sources of the infection. I think you all know these sources of the infection. The first source is wild animals. It could be mosquitoes or like dogs or any wild animal, because a lot of them carry inside some diseases which are very dangerous for humans. Another source is farm animals, because farms have a like very huge density of animals like pigs or cows or like goats, and these farms can be source of potentially harmful bacteria or other diseases. And the second or the third possible source of infection is just people, because it could transmit from people to people. There are different ways of spreading the infection, right? The first way is airborne, so infection could spread through the air, or it could spread through the direct contact from human to human or from animal to human, as we had with coronavirus. As you know, yes, it was originated from animals, and as many human infections. And the most important for us for today, source of like and the way of like of spreading the infection is infected water, right? Because this type of spreading the infection is I don't know how to say, it's uh, very we can control this if we treat water right. Yes, it's very difficult to control spreading the infection through the air, but it's much easier to control spreading the infection through the water. Moreover, wastewaters could serve as indicator of spreading the infection. For example, after the pandemic of coronavirus and in progress of pandemic of coronavirus in Moscow, we have very strong correlations of the amount of coronavirus in wastewater and the amount of people who is sick, who is hospitals right now. So and even though we can predict if we use wastewater as an indicator, we can predict how much people will get very sick in a few days. It's very interesting information, and I think our project will work with this as well, because if we can do this with coronavirus, I think we can do this with other infections. So if the main source of like infection for now for us for today is infected water, we should speak about wastewater treatment and disinfection in different types of wastewater treatment systems. You know, after each like domestic or municipal wastewaters, they should go to, or maybe agricultural or industrial wastewaters, they all before they can be discharged to nature, they all should be treated and purified from some like pollutants, from biogenic elements like nitrogen, phosphorus, and other elements, and they should be treated and cleaned from xenobiotics, some like complex organic molecules, which we all like use in our life, like pharmaceuticals or pesticides, right? And also it's very important to purify wastewater from possibly pathogenic organisms. And different wastewater treatment systems not so well like perform this task not so, with different efficiency, you know, because activated sludge system, you know, it is very popular system in Russia and a lot of countries. Its system includes biological treatment, but the retention time in wastewater treatment facility is very low. It's about 6 hours. So biological disinfection is not going very well in these types of system. Treatment lagoons and constructed wetlands are different types of wastewater treatment systems, and they include a biological disinfection in much higher value. So we will talk about that a little bit more. Uh, there are different types of disinfection in wastewater treatment facilities. As I told, in conventional treatment systems like activated sludge systems, the biological disinfection is not going very well. So people use some chemical and like physical disinfection systems, like ozonation. It's treatment of water with ozone. It's O3, which is very strong oxidant, and it kills a lot of bacteria and viruses, and like successfully purify water. And ultraviolet irradiation, because this ultraviolet radiation is like high energy radiation, which are often used to disinfect wastewater. But these types of disinfection are not very effective, because there are some forms of microorganisms like spores, which are very stable and have a strong resistance from ozone and ultraviolet radiation. so they can go through these types of wastewater treatment and go to the natural like surface waters like rivers or lakes, and some people can then drink that water, which is not very good because it can include some possible like some dangerous bacteria and pathogenic microorganisms. Biological disinfection is going very well in constructed wetlands, and it is the most efficient, but it depends. For successful biological disinfection, it needed a long time. It needed time about 5 or 4 days of retention time in wastewater treatment facility to purify water from all possibly pathogenic microorganisms. After any wastewater treatment, we usually have some microbiological control. But the conventional methods of microbiological control are not very effective, and you will see why. The first method is just sowing on a petri dish. So you can take a sample of treated wastewater at the outflow from wastewater treatment facility, or agricultural object includes agricultural wastewater, and it just sow it on a petri dish and see with nutrient media, of course, which allows some bacteria to grow, and you can see how much bacteria grows, and you can evaluate how much the total number of bacteria in the outflow. It's good indicator of microbiological pollution, but it's not very direct, right? Because it's not, it doesn't evaluate pathogenic bacteria. It is evaluate overall bacteria and only cultivated ones. The second method is coliform index. It's very well known and often used for drinking water. This index, it is a method of sowing on petri dish as well. This method includes bacteria from family Enterobacteriaceae, and it is an indicator of fecal pollution and general microbiological pollution. And the third method is direct calculation in special chambers, when you can calculate and measure the total number of the cells in the treated wastewater. It's the overall number of the cells. Sometimes this method includes some special stainings, which helps to calculate, or you can just directly calculate the cells through in the light microscopes. All these methods has some general flaws because they analyze bacteria in general or cells in general, this bacteria does not include pathogenic species because it's dangerous to work. It's dangerous to produce some and evaluate some pathogenic bacteria directly. We have to make special laboratory for work with pathogenic bacteria. We have to buy special equipment. We have a lot of like restrictions and rules how to work with bacteria. It's dangerous for human and require many skills and professional people. So nobody detect with this direct conventional methods. Nobody detect pathogenic bacteria directly. Also, viruses never included in this analysis because you cannot grow viruses on a petri dish, right? You cannot analyze viruses using these methods. So you skip any danger from viruses if you're using for microbiological and sanitary control only these methods. And these methods have a large measurement error, because most of the counting of cells is performed by the human eye, and we make a lot of mistake when we count single cells under the microscope in a petri dish. On the contrary, we have modern molecular methods such as sequencing, especially new generation sequencing or high throughput sequencing. It's 16S RNA Illumina MySexis sequencing and quantitative PCR or quantitative or real-time polymerase chain reaction. These methods, their main feature is they do not depend on cultivation. These molecular methods work with DNA instead of working with living cells. so you can detect pathogenic organisms directly. Also, using these methods, viruses could be detected because you shouldn't have to cultivate anything. You just extract DNA from your sample and you can analyze viruses as well. And molecular methods could be quantitative. So we can evaluate the number of pathogenic cells in any sample which we can take. So these methods are more suitable for our project, and these methods are um, very direct and very precise. The general workflow in our project should be as you can see it on the slide. First of all, we should collect samples from any chosen object. Then we should extract total DNA from this using any protocol. We will choose the protocol later. And then we have two possibilities. First of all, I recommend perform sequencing, 16S RNA sequencing, to detect all microbiological components in the sample. We will detect all bacteria and archaea in the sample, and then we will see what are the microbiological structure, and we can see how many different pathogens we have in our sample. After that, we can perform a quantitative PCR reaction to evaluate directly and precisely each pathogen we will choose. After that, we will have number of pathogenic cells, and using this information in different sites, maybe downstream from the object in the rivers or in natural objects, which can have discharge of water from wastewater treatment facilities. We can use this information to evaluate how dangerous this waters to people, and maybe even offer some measures to make this wastewater treatment facility and waters around them safer for people and animals who lives alone. Okay, so let's talk about this more detailed. First of all, we should collect samples for our study. For that, we should pick up an object for study. It should be wastewater treatment system, wastewater treatment facility of any kind, or agricultural object like paddy soils, rice fields, or something like that, which use a lot of water and have water discharge. This object should be important for territory. These waters should be in contact with people, with maybe some villages who use this water for irrigation or even for drinking. Then we should locate places of water discharge to the lake or to the river we will choose. And on that places, we should take samples. As the samples of sediments or the soil, if it's lake or river, and samples of water at the input and at the output of treatment facility or agricultural object. We should take samples around water discharge and downstream, because we want to see how far the pollution goes from wastewater treatment or agricultural object, how far downstream we can detect some pathogenic or possibly pathogenic species of bacteria. After taking, samples should be cooled to 4 degrees Celsius and transferred to the local laboratory in your country as soon as possible, because right after we take sample from their natural environment, it starts to change. We change the environment we change the environment, and then the bacteria should change. Some bacteria die, some bacteria should reproduce, and so we will not have the natural picture in 2 or 3 days. The picture will be different. So we should very fast to cool. We should very fast cool down our samples to 4 degrees, and then transfer to laboratory and extract DNA. DNA should be extracted always as soon as possible to adequately represent the sample. After the sample is taken, it starts to change, as I already told. We extract from the sample total DNA. We have some protocols for that. For us, we choose Power Soil DNeasy Power Soil kit. This kit allows to extract DNA from a huge variety of samples, and it suits for soils, it suits for sediments, for some sludge deposits, and we even have techniques how to apply this DNA extraction kit to some water samples. So we will use the one extraction kit for all types of samples to have compatible results. After DNA extraction, after DNA extraction, uh, sorry, yes, after DNA extraction, all nucleic acids should be frozen because after further analysis we will perform in Moscow, and these samples should be transferred to Moscow. I will tell later about that. And to properly keep them and to not decompose DNA, we should freeze the samples at temperature minus 18 degrees Celsius all over. It's perfect to have minus 28 or 32 degrees Celsius, but minus 18 in like any home freezer samples will be safe. Okay, the next step is sequencing. We want to evaluate all bacteria which we have in our samples, and we will perform 16S rRNA sequencing. We will read only one region of DNA, its 16S rRNA gene, but this region will help us to detect all bacteria because we have big databases which have information about this region in a lot of different bacteria, including pathogenic ones. This data will help us to detect all pathogenic bacteria we will have in the sample. We should remember that this analysis helps us to detect, to list all bacteria we have in samples, but it is not quantitative. We can use this list to then perform direct quantitative analysis of bacteria which we will choose. And for that purpose, we will use real-time or quantitative PCR reaction, so-called real-time PCR. Using data from sequencing, we will determine a set of microorganisms for quantitative analysis. One reaction of PCR allows to detect only one microorganism, but it's very accurate analysis. Also, it allows us to detect viruses, include coronaviruses or any other viruses which we can pick for our analysis. And we should remember that each reaction here is quantitative. We can calculate how many pathogen cells we have in our sample, so we can evaluate how dangerous this sample could be for human. Is it relatively safe and it will be decomposed in nature, or it's very dangerous and humans should not interact with that water? We can make some conclusions like that using quantitative PCR reaction. So this is the general workflow for our project for overall. And here I have some notes. So collection of samples and total DNA extractions should be and will be performed on site in a local laboratory in your country. All necessary equipment we will provide in our project. For now, we are in the progress of buying all necessary equipment for collection and DNA extraction. We will buy DNA extraction kit like Power Soil DNA extraction kit, and all like tips, pipettes, Eppendorf tubes. All you need to extract DNA and keep it. Then these samples of DNA, they are already stable. They will decompose very slowly, and the how to say proportion of different organisms in these samples of extracted DNA are already stable. So these samples will be transferred to Moscow altogether, and on special equipment such as amplificator of nucleic acid and sequencing machine, we will perform sequencing, analyze this data together with you, and then pick up then we will pick up a list of pathogen for each country. I should note that in each country, on each object, it should be different. It could be different different pathogens, because different countries, different climates, different zones, different wastewaters. So number of and list of pathogens could be different. After that, we will perform quantitative PCR reactions and analyze how much pathogenic cells we will have in each samples. And then we will all together analyze this data and make some conclusions about how dangerous these wastewaters on that object you choose, and what we should do to make this object safer. So I think that's it for the workflow of the project. I think this general idea of what we want to do, and after that, I want to have two more online seminars about the methods we will use, because people who attend this meeting will work, I think. Yes. Yes, I want to make an online educational seminar to describe in details how DNA extraction works and how we read DNA, how 16S rRNA sequencing works, why we chose this region to determine all bacteria and also archaea in our probe, how it works, and how to analyze data from these methods. And after that, I want to have one more seminar about what is quantitative PCR, what is polymerase chain reaction, and how it works, and how we can use it to detect pathogenic and in general any microorganisms we want. So this is our plan for following seminars, and I will inform you about the dates, about where and how it's gonna be later. After that, I think it's gonna be in a month or two. I believe that some restrictions due to coronavirus will fall and will end, and we will have a possibility to make offline seminar with training with DNA extraction and working with laboratory equipment. So yes, I think that's it for today. That's it for general part, and it's time for your questions. If you have any questions about the project, about your object, about anything, please ask, and I will try to answer this question. I will stop the presentation. Hello, everyone. Okay, so if anyone have questions, you can raise your hand in Zoom. You remember how to do that, and just ask me a question. I'll wait for a few minutes. For now, I cannot see any raised hands. Yes, yes. Professor Hai, I can see your raised hand, but Nishesh, can you please give oh, I can hear you. Hello.
Hello, Sergey. Thank you, everyone. Hi, everyone. I have a questions about the project. So thank you for the presentation. And I know that in the project, you will take the sample from different country or from different environmental component. So can you tell more about the type of sample that you will focus on, and from this sample you will isolate some specific microorganism for the specific purpose of project. So can you tell us about the scale or the scope of the project in terms of microorganism sampling and analysis? Thank you.
Yes, thank you for the question. This is educational project to spread the knowledge about molecular methods and how they work with microorganisms in different environments. But because of current situation, we think it's important to focus on analyzing some possible and pathogenic microorganisms, because there are a lot like dangerous places in the world which can be a source of such microorganisms. So each country should pick up an object and take samples. It could be samples of sediments or soil, which could be infected, or it could be samples of infected wastewater or treated water. Yes, and we have protocols to extract DNA from any kind of samples. And after that, we will analyze all DNA, no matter what the source is, in the same protocol, because it's suitable for any source. After that, we will describe all pathogen microorganisms we will find, and then pick from them the most dangerous ones, and evaluate precisely them in the water on the object, because we want to understand how dangerous these waters, which you will pick, for the people who live around them. Yeah, thank you. I think I answered. Any other questions? Nishesh, I think we can allow people to just turn on their microphones because it will be easier to ask questions. I think all people can turn on and turn off the microphones. So if you have any question, just turn on your microphone and ask. Okay, so if we don't have any questions about this project, we can finish our meeting. I just say some concluding words. So today this was general seminar about what the project is and how it's going to be performed, the general workflow. And the next seminar will be, I think, in a few weeks. I believe it in 2 weeks, when I will describe in detail DNA extraction protocol which we choose. DNA extraction protocol which we choose, and why we choose that, how it works, how you can work with it, which equipment you need to work with it. I will describe it in details, and we will speak about a little bit more about sequencing, how it applies to our goals. This is going to be next seminar. Okay, I think Nishesh will share all information about this seminar, and we will share the agenda through the email as we did last time. So if no question, we can finish meeting. Thank you everyone for attending the meeting. I believe I will see you all on the project. I will see you all online, and we will make some interesting research.
Thank you.
Yeah, thank you.
Thank you very much.
Yeah.
Bye bye, everyone.
Yeah, goodbye. Goodbye.
Okay. Thank you.
Long time no see.
Spasiba.
Chào anh.
Cảm ơn.
Yeah.
Okay.
Yeah, goodbye.
Thank you, everyone. Thank you.
Thank you, everyone, for attending the meeting.
Thank you.